then the button is you got to hold it down and the light will come on from the regular microphone. Also?
Okay, cool.
Yeah. All right.
Cool. All right. Everybody, my name is Mark Harris. I'll tell you a little bit about myself kind of before we get started here. Am I in a good spot ish? All right, cool. So October this year, I guess will be 20 years for me in the beer industry. I started at Sweetwater, driving a forklift and loading trucks in 2003. I'm a mechanical engineer by trade, so the doctorates in microbiology that are here, please forgive me. I'm going to do my best. So yeah, I started it in Sweetwater in 2003 and kind of led into production management and operations there, and was worked down there for about 3 years. And I left there after having my first kid and went out to Athens, Georgia, and I designed and built Terrapin for John and Spike and operated that facility as their plant engineer and production operations manager for 5 years. Real small company at the time, so obviously we all wore a lot of hats. I did a lot of the distributor planning and production planning, ordering, all that kind of stuff. So yeah, that was kind of the first 8 years in production brewing for me. Learned a lot from all those folks, and I took up distilling as a hobby in the back of that brewery. And then one day John said, this is probably not the right place to be doing this. And I was like, well, yeah, that does make some sense. And so from there, I left and I was the Eastern director for Pro Refrigeration, a glycol chiller company, for 3 years. And so I designed a lot of breweries for a lot of folks and kind of helped with, throughput, getting your grain in one side and the packaged product out the other, working for that company and traveling around. Got to go visit a lot of breweries and wineries and distilleries all around the country. So after 3 years there, the last 8, I worked for Craftwork Brewing Systems out of Detroit, Michigan. So designing heat exchange surfaces on tanks, overall tank geometries, did a whole lot of different things there, made some really interesting things. We built turnkey brew houses for quite a lot of breweries, and so I'd go to site and kind of help people start those up and do overall whatever they needed in the industry. So size boilers, chillers, heat exchangers, pumps, valves, fittings, design control panels, and I studied with Siemens and did their whole brow mat program, so I'm a pretty decent computer programmer overall. And I'm really happy to be a partner in Midwest Microbio. I'm not here trying to necessarily drive sales down your throat, but more to teach you about PCR. So I am the full-time director of sales for Midwest Microbio. So if it does come off that way, I apologize. That's not my goal, but it is my goal. So I brought some stickers and some cards if you guys want to pass those around, if anybody wants that stuff. Who knows what PCR is, or q-PCR? Just show of hands, just kind of wondering how many micro folks we have. Okay, cool. That's awesome. So, yeah, anybody feel free to take a card or one of these. And who's running PCR or q-PCR at their brewery now? Awesome. All right. PCR, this is kind of the objective. This is the main sheet. So I'm going to try not to read the slides to you guys. I know you can all read. We're going to kind of go over PCR, q-PCR, what it is, the difference between the two, chemistry, process, and how it's used in the brewery. This is a pretty dense slide, and basically polymerase chain reaction is what PCR stands for. Everybody heard them talking about it when they're saying, hey, I got a COVID PCR test, right? So there's a few different things that are basically the parts of the system, right? So your DNA sample, if you were, COVID, then you'd be looking at your swab sample. We're pulling either a can sample into like a 50 ml tube or something off a bright tank, the end of fermentation. So a small shot glass 50 ml vial of beer is our DNA sample. That's what we're testing. What we're looking for is microbes, anything that's alive in the beer, and what we're doing that so that we have quality control and know the beer spoilers, the things that can ruin your beer. Worst case scenario, making the cans explode on the shelf and whatnot. So that's your sample, your beer that you're going to test to try to figure out what's in it. And then the primer is something that you get from one of the PCR companies. And each primer is a complementary RNA strand to DNA that has the exact DNA sequence of what you're looking for. So if it's Brettanomyces that we're trying to test for, then it has the complementary Brettanomyces DNA. So it basically, in this reaction, you're trying to look for one specific spoiler in each well. There's also something called multiplex, but we'll kind of try to stay on the lower level of that. So nucleotides, everybody kind of probably already heard about C, G, A, and T. Those are the basic building blocks for DNA. So those are, what all DNA is built out of, right? So we take this sample, we shear it and break it apart, digest it, and the DNA that's there, the Taq polymerase is a bacteria that was discovered in Yellowstone in one of the thermal pools there, the hot water. So it is a heat-loving microbe, and it's a pretty amazing one. So it actually, in conjunction with the primer, grabs a hold of the exact DNA on the strand in your sample, and then basically kind of goes down and replicates that, right? So if you have a very small amount of something that would make your beer go bad in there, this kind of goes through, finds that, and goes and replicates it to a point where we can actually detect it. So super low level, kind of taking all those nucleotides out of this solution. The solution is the next kind of key, the mixed buffer, caustic solution that allows for this reaction to take place. So kind of breaking down some of the DNA to allow those nucleotides to be available. The Taq polymerase kind of jumps on and takes it from there. This middle part is the 3 steps. Denaturing is kind of where you break apart the DNA from double-stranded to single-stranded. Annealing, where you kind of see right here, that's where the Taq polymerase will actually match up and grab a hold of that DNA sequence. And then the extension is it pulling the nucleotides out of solution into that exact sequence and replicating it. So each time you heat and cool, that's what it does. The chart over on the right-hand side, it's the temperature cycle. So each time you cool down and heat back up, you create more of those copies. So if you start out and you only have one copy in the very beginning, one little bit of, let's just say Brettanomyces, because that's the one I'm picking to talk about tonight. So on the right-hand side, you can kind of see after 20 cycles, you have over 1 million copies of that as it doubles each time. Yeah, that's basically the skinny there. PCR versus q-PCR. So with PCR, you're kind of looking a lot of times at like a little cartridge or a pregnancy test. But bottom line is with PCR, you get a yes or no. Is there some of what I was looking for in this sample or not? So yay or nay. Q is quantitative. So in quantitative PCR, the cycler pulls a lot of data, and as the fluorescence increases, it runs statistical analysis, and you can kind of back into how many cells per milliliter you had in the beginning. So that's kind of the difference between those two. Are there any questions so far? Okay. So as this reaction goes, your baseline is kind of the very first part of the reaction. That's where it's still kind of negligible, the amount that you have. And that's where the Taq polymerase grabs a hold of that DNA and starts to replicate it, but it's not enough to where we can measure it yet. And then you have the exponential growth phase, linear growth phase, and then the plateau. So basically, as it kind of goes through, when it kind of comes out of the negative area down the bottom where you can't really tell how much you have, then that's the case. I'm not going to dwell too much on this stuff. So overall, there's a couple different kinds of dye overall used in the industry. This is intercalating dye. So TaqMan is the one on the right here. And when that dye, it actually goes into the DNA and takes the place of what was there. So it kind of kicks out the DNA that you were looking for. And so that's kind of like a glow stick. Once it's there, there's no way to stop that from fluorescing. So with TaqMan, once you kind of find what you're looking for, it stays there. Our system uses cyber green. Cyber green only bonds to double-stranded DNA. So using that is allows you to denature the sample at the end. And you heat it up to the point where the DNA actually disassociates and goes back into single-stranded DNA. And that allows you to do a melt curve analysis. So as you can see, kind of at the top, I'll point at my computer like you guys can see that. Up at the top, as you heat it, the temperature is going to the bottom, and it comes down, and then you see that kind of where you're coming from, double-stranded DNA to single-stranded DNA. So that'll stop fluorescing and give you a baseline 0 read at the end of it. So all the different DNAs and primer sets will disassociate at a different temperature. So doing that allows you to say, hey, I actually got what I tried to get. I know that this is what it is. You can also tell a bit from the peaks. This is the first derivative of that data. So you can kind of identify between, like for HOR ABC, whether it was the HOR A gene or the BC gene, or if you're looking at a diastatic yeast, you can tell whether that yeast is highly diastatic or poorly diastatic. So a lot of data that can be derived from that melt curve analysis. Definitely something to be looked for. And the systems do all of the statistical analysis for you. We've kind of programmed all that in there. So this is our result for SIR yeast. So basically, it kind of kicks out, this is 100,000 to 500,000 cells per milliliter. With Stay Plus, you have 1,000 to 5,000. Brett all the same, and no HOR ABC in the sample. So you kind of see as the different primers that you put into a different vial that went into the cycler. Each one of those is one of the different curves that amplified through there. Does that make sense? Any questions? Sweet. We started this company out with about 16 assays. Now we have 43, and many more are growing and coming. We've had folks that kind of reach out to us and ask us, hey, we're kind of trying to figure this out. Is there something we could come up with that would would target this or that and the other? So we're all open to that. And so, like I said, we've kind of grown the amount that we have from 16 to 43 at this point, and growing. So there's a lot of inhibitors to the PCR reaction. Coffee's a really good one that kind of keeps it from being able to snag the DNA and replicate well. Kettle sours are a pretty tough one to deal with. Yeah, some of the new styles out there with a lot of puree and those kind of things, they can inhibit that. So your lab bench technique to process those samples is really important. If you do have an inhibited reaction, then you have to further rectify that DNA to make it available and usable. And our system also does statistical analysis to tell you at the end that you had no inhibition. There are also positive controls that are built in as well that you can put in, and they're not the exact microbes that you would bring in and contaminate your brewery, but they contain some of the DNA from that. So you can put those into the vials as you run your sample, and it'll come up with a positive control. So you know that, hey, everything I've prepared is correct and working. If I put this in there, then I get a positive. Cool. So ideally speaking, you don't get a whole lot of these in your beer. This is a kombucha sample. So it gets kind of boring if you're really good at brewing, because nothing ever amplifies, right? Yeah, Joe. Yes, absolutely. Like our system, we have 16 different protocols that you follow. So it's just a little bit of a different step one way or another if you're dealing with a stout or a fruited kettle sour. Some of the systems are kind of based on German beers overall, and they can get kind of muddied up if you're doing that. So you get false negatives, and obviously if your laboratory folks are giving you false negatives, then we're all pretty upset overall. Our put out bad beer and our company goes out of business and nobody likes the brand anymore. So does anybody know about VBNCs, viable but non-cultural organisms? If I get a show of hands. Okay, cool. Basically you're going to miss these if you're just doing plating. Okay. So viable means it can be living in the beer, and if you culture it up on a plate, it's not going to go up like you want it to. If you are, an amazing microbiologist and you do anaerobic plating, it's a very intensive process, takes quite a bit of time, and there's a lot of organisms out there that'll take 3 and 6 months to grow. And so in a 2 or 3 day micro hold at the end before the beer's going out, you're not going to pick up those things. PCR between an hour and a half and about 4.5, 5 hours, depending on which system, you have quantified results of the exact microbes that are in there. So that time frame really helps out in modern brewing a lot. The yeast suppliers do from time to time give you infected yeast. So being able to bring that stuff in on the dock and test it before you put it into a big fermenter of beer is really nice. So being able to kind of take that and have a result in an hour and a half. Same kind of thing on a canning run, right? Like you can go out there and pull some of the first cans off the line, and if the CIP person came in at at 6 and they had the line up and running at 8. And, you know, you can pull samples off there at like 8:15 and have good results by 9:45, so that, hey, this beer is clear, like it can go to market and on the truck that day, right? So those are kind of things that, you know, this technology is fast. It used to be cost prohibitive for breweries to have, and now as things have developed, it's a lot more approachable for folks. So sensitivity and selectivity, it's basically going through, you know, a yeast pitch where you have billions of cells in a milliliter, which is really hard to really think about if you kind of look at it that way, like billions of cells in there, right? And then you can actually target the 1 or 2 cells that were in that milliliter of liquid, and you know, pick that needle in the haystack out and say, hey, you know, here we are. If we have, you know, 1,000 cells per milliliter of this, like we're in pretty big shinola. So it's nice to be able to use these DNA tools to identify the things on this level. This is kind of the benefits of that, right? If you're plating, then you're getting a yes on general bacteria. But do you know if that bacteria is hop resistant or not? You don't, right? There's no way to know that. So these are all the kind of, you know, yeses and nos of plating versus what you get when you're doing PCR. So diastatic versus non-diastatic yeast cells, right? Like you're not going to be picking that up. pH tolerant resistance genes, again, the viable but non-culturable organisms. Lactobacillus acetotolerans really has ruined a lot of barrel-aged beer for folks. There's a huge recall for that. And, you know, whether you have phenolic off flavor producing yeast or not, right? Like whether it's positive or negative for that, that's something that you can do with PCR that you can't with plating. And then if you're doing like LCSM, whether you want to know if something's copper resistant or not. Okay, so this is a myth. Extracellular DNA, eDNA, viable cells. There's some kind of rumors out there that PCR is picking up a lot of extra DNA that's in the sample. So the first step in the process is centrifugation. So you have this little 50 ml tube there, and here we've got live yeast cells, a dead yeast cell, and then some of the DNA that's kind of floating around in there from other dead yeast cells. So we're looking at beer. We're kind of starting to test this at the end of fermentation, you know, maybe before flash pasteurization, after flash, before and after filtration in the bright tank, going into the canning line, before and after a tunnel pasteurizer. It's a perfect tool for dialing in a tunnel to decide, hey, we're coming in at this microbe level, and if we run this many PUs on, say, like a milk stout, right? All the PU levels and how far people are cranking up their tunnel pasteurizers were all kind of decided on German beer. An adjunct pastry stout takes a lot more PUs to get it clean than it does a Pilsner, right? So first thing you do is spin this in a centrifuge. So you got your DNA in solution up top, and these next few slides kind of confused me a bit when I kind of first saw my partner put them together. And so basically we're adding one step to the right on each of these going through. So basically we have 1 and then 2 and then 3. And so as we go forward in these slides, it's like, okay, this is still our original sample and it'll go across. Hopefully that'll make it a little bit more clear. So we centrifuge all the lighter stuff, the cells to the bottom, leaving DNA in solution. And then you pour that beer off, leaving you with your cells in the bottom. Next step after that is adding in the digest buffer, right? So that's the chemical that dissolves and does that so you can see, I want to point down there. So you can see, here's our cells that we spun to the bottom and dumped the beer off. And then you add your digest buffer in, and then you've got liquid again. And that's where you start to lyse your cells. That means you break all the cell walls and leave that DNA available to be grabbed. That's where you're putting all those other nucleotides into solution, so the Taq polymerase then has them there to grab, to put on, to build the chains that we're looking for. And then it goes into the thermocycler. And so that's where it amplifies, cools down, heats up, cools down, heats up. And each time you're doubling the amount that you have there until you have a detectable level. So centrifugation, you can't spin DNA out of solution, is the real takeaway there. So if you hear the rumor, hey, people are, saying, oh, we're picking up dead stuff, that's the case. And then your DNA sample to add to the PCR reaction for amplification, you just get the DNA that came from the live cells that we were looking for. I kind of already talked about this a little bit. Here's a couple microbes that picked up before pasteurization, and then after, you can see a clean sample. So this is something going, through like a flash or a tunnel is basically what you can look for. Total time, I already talked about that too. I kind of get ahead of myself sometimes. So we're almost done, everybody. We're doing good. Hour and a half versus a couple days. Some of the bigger systems and bigger cyclers and stuff take 4 or 5 hours for PCR. 20 to 30 minutes kind of active lab time. And then you're letting the cycler run the cycles for an hour and do that. There's several different systems out there. So a lot of them are kit based, and that's something that people have kind of come up with and reserved for the brewing industry, because they think we can't handle real lab work. So they basically kind of taken all the pieces and parts you need and kitted them up, and they sell it to you on a per use basis. So this kind of looks at, our system, and as you do more and more sampling, the kit based ones really add up. So 2 is